Good evening, ladies and gentlemen. It's nice that you've found your way here, despite the hot weather today. Now, first, I would like to explain something about the context in which this event is taking place, or took shape. Last semester we had a sequence of talks which was called Board Research and Consequences on Society, and that was about the influence on industry on research in various fields, and more precisely in the area of biotechnology. We expressly do not want to question research in laboratories. We are talking about the application on food production and agriculture. And because the food supply is what we are talking about, this is something that affects everyone. It's not something that only a small, selected group of people should debate. It should be a very wide debate. And that's exactly what we want to do tonight. I would first of all ask Professor Schiemann to give us his presentation. Yes. Thank you for the introduction. Because the introduction talked about the fact that industry has an influence on research, I would like to start by saying that research needs third-party funding, but we do not take any funding from industry. These come from the German Research Society, from the European Union, from government ministries and such. Some time ago, I went through the city where I now live in, and I was quite surprised to see in one of the windows, or two windows actually next to each other, more jeans. If you then looked at the following windows, this continued as generation house. So, to continue that word, if you have more genes, it leads to generation houses, and that is a nice view on the debate that we have today. Because the consequences or the usefulness of certain technologies, when discussed, should be viewed in the context of what we have done for about 10,000 years in the context. If we regard those different technologies with which we can change the plant genome, I'm sure you would agree that none of these technologies is safe per se. But we have got used, since about 10,000 years, to say we have a history of safe use. So we know that even the things that we produce with classical procedures and the foods we produce from those are not safe per se. Just think about different allergic reactions that can be caused, where some plants have to be treated in some way, say by cooking them. Which means that the question is, does it make sense to look at the product, which is a genetically changed plant, in terms of the procedure that's being used, or in terms of the result, the properties that have been inserted in and then look at the various plants on a case-by-case basis. With these new technologies, in particular genome editing, there is of course the question GMO or not GMO, a typical Hamlet-type question, or the Faustus question: what is your stance on regulation? Should these new plants with their new properties that have been produced by genome editing regulated as genetically modified organisms or not? If they are regulated as genetically modified organisms, then they underlie strict security assessments, so few people will be able to afford to use these technologies. And then we have what is called democratic technologies, the CAS technologies, which smaller and medium-sized breeding businesses will be able to use. So, just to compare, you need about 15 to 20 million euros to produce a plant with a new property using genetic engineering and bring it onto the European market and also in other regions of the world. A safety assessment on that is also linked to a very high cost. If we regard the question whether to regulate or not, which kind of safety assessment, at which level to use, we have the full scale or the reduced scale that we could use. If we look at this on an international comparison, then we see that in some regions of the world there are plants that in Europe perhaps are regulated as GMOs, but not elsewhere. So US, Canada, and also Argentina, Brazil have meanwhile found quite different systems to regulate these products. And in a workshop with Chinese colleagues that we had a few weeks ago, we were told that we have gene editing with CRISPR-Cas. We use this on maize, and in particular CRISPR-Cas, which is genome editing, is used across the world. All right, now what's the picture in Europe? In Europe, in 2007, there was a commission where the so-called competent authorities of the European Union member states, sent 2 experts each to take part in that commission, and that commission in 2012 had laid forward produced a report which hasn't been published until today, but it has been circulated, and this led to a number of conclusions why certain technologies, why certain plants should be regulated as GMOs or not. And to just point to an example, many of you will have heard the story of the cibus rapeseed, which is a rapeseed that has been produced by genome editing for herbicide resistance, and the company behind it contacted 6 competent authorities in 6 member states, asking them whether that is a GMO or not, and all these authorities said no, this is not a GMO, because this rapeseed, which was produced by this technology called ODM, cannot be distinguished from rapeseed that will be produced by other methods such as radiation or naturally mutated rapeseed. The German competent authority, the Federal Authority on Food Safety, stated its position and also said that this is not the GMO in their view. The procedure hasn't started yet, but it's being prepared, and the European Commission in June of last year promised to find a legal clarification until the end of the year. Now that has been postponed to the end of this year, and I assume that even till the end of this year we will not have this. Now, what is important is that member states on all levels have been debating this, and for once from the legal perspective, but also, of course, from a scientific perspective. And in Europe, of course, we can expect that no matter what the European Commission will decide, it will be brought to the European Court of Justice. So in Europe we will have to wait several years until we have this decision. Now, in my view, it's correct that we have to meet the global challenges that we all know, and for that we need a broad range of tools, and all instruments of that toolbox should be used, providing, of course, that we do not make any stupid things. So any technologies we use, including Cas or genetic engineering, have to be compatible with consumer interests and the environment. And the same conclusion goes to GMOs, affects GMOs. There was a Commission for Agricultural Biotechnology in Rome, and the conclusion and recommendation was that the FAO, that organized this, concluded that the problems we have to solve regarding food safety and providing nutrition have to be solved with including all technologies, and that the potentials of genome editing in particular, CRISPR-Cas, are very important, and that in the future they could be used. And I remind you of an interview that the director of the Swiss Organic Agriculture Institute had with the Zeit newspaper, in which this man, who was labeled an eco-pope, the director of this institute, very clearly said that he could very much imagine genome editing technologies being used for the purposes of organic agriculture. And later, in the context of a TV program, he saw himself quite surprised about the shitstorm that he had faced, the violent opposition, and he was interested in starting a debate on whether these technologies are useful in organic agriculture, and I think that is very sensible, and it should not be ending in that strong opposition. So that's what my current thinking is. Thank you, Professor Schiemann. And next I would like to call on Ricarda Steinbrecher to give us her perspective. Now, I am a biologist, a geneticist, and have done a lot of work in regular genetics and gene therapy. The interesting thing was that human genetic therapy in the 1980s said 10 years at most, then we'll have all the solutions. And in the mid-90s, we were just as far as we are in 2015, which means that if we had a certain statement, we made that statement on the basis of the knowledge at the time. But of course, we only had a very limited knowledge. And the idea that something could be done didn't quite match with the facts, and I learned a lot from that about the opportunities that are being stated regarding plants and what the possible effects could be. I would always put a large question mark behind that. The important thing for me was to evaluate safety, risk questions, because you can't do anything if that means that the patient might get more sick, or that the germline is being damaged. So that's where I'm coming from that kind of concept. And for years now I've been following the UN Protocol on Biosafety. I've been part in various expert gremiums, committees, and risk assessment is my expertise. So what shall we do with these new technologies? Because those, as Professor Schiemann said, are debated globally. And how do we deal with the huge, these very rapid developments? Is it not just whether or not it's GMO, but what you can do and what the possible problems are? So that's what I will now deal with. Just shortly, these new breeding techniques, which is the way they are often called. Mostly we talk of new genetic technologies, but new breeding technologies is a term that should be regarded, and I've tried to bundle this into 3 different groups. The first group is genetic and engineering and then genome editing, the oligonucleotide-driven mutagenesis, ODM, which was used by Cebus for the rapeseed, which is sometimes regarded as a GMO, sometimes not. But that is not the main question. I'll start from the scientific point of view, not the legal classification. Now, this whole area, these are procedures, methods that work as molecular scissors that cut DNA in certain places and then cause changes by using repair mechanisms of the organism itself and activate those mechanisms. So these are doesn't mean that there are precise results, but precision is certainly being increased. I'll come to that later. So that's number 1. And number 2 are the various conventional genetic engineering procedures, which now sometimes are divided into by their origin into cis and intragenesis. The gene, whether the gene comes from a species with which it could also be combined in conventional breeding, at least theoretically. Other methods, which I will not go into, but there was a paper that I wrote on this. I don't know how far this is present. We could include this on the website later. It is in German, explaining all those methods. There are a few copies in the room. But mainly I wanted to talk about genome editing and genetic engineering. So the question is, what is new? You can see here, all these things are possible with CRISPR-Cas, which is this very new technology that has only really been taking shape 2 or 3 years ago and is now being used in research very widely, which doesn't mean that it's very widely used in application or in things that are being circulated. Not that, but in research it is very widely used because first it is very cheap, it's very simple, and it's very quick. So some people say, well, why don't we just give this to farmers themselves, let them apply this themselves, and that then is the absolute democratization, and that of course is the question, because I will come back to what you can do, and that should be regarded. So the thing that is new is an increased ability to have deeper and more complex changes. So really far-reaching, take this gene on that and change it and produce very completely new kinds of metabolism, and that's what you can do with Cas, and that area is called extreme genetic engineering or synthetic biology. And the second thing is the change of certain targeted DNA sequences, which is called precision, but I put a question mark behind that, which I will come to. So to switch on and off, or inhibit or change or insert genes, but also on a very different level, which is called population scale genetic engineering. So populations, whole subgroups, a whole species could be affected. CRISPR-Cas could be put into a method which is called gene drives, which kind of divert the whole classical rules of inheritance that we know, and guarantee that a certain gene is always propagated to the next generation. So the whole population, the whole species could be changed, and genes could be switched on, switched off, or strengthened or weakened, and that is quite well progressed. But of course, there's a high need for debate, and scientists around the world ask for the debate to go on, ask people to participate, because it is not just a discussion in science, the question how far we should go and where the limits are. So this idea of precision, in terms of precision, normally you'd say that the idea of precision means that you know what you're doing. Now, this, and I'll come back to that, this is actually not really possible. You have precision at the nucleotide level, but that's just the bottom level. You have various levels which you have to consider. What's missing is the contextualization of what's called the epigenetical landscape. And then we have the organism as another level. We have the socio-economic situations that changes across the world. We have to say that precision at the nucleotide level, if you just look at that, that gives you a false sense of predictability and safety, because there is no data that could support such a conclusion. It doesn't mean predictability, and doesn't also mean safety, at least not automatically. In some cases, this could be given, but not in others. What is important to consider here is the predictability or the unpredictability, because there are what is called off-target effects. So you may have changes not just where I want them, but in different places as well, which are not intended, because there may be similar marker sequences. Even if I change just one nucleotide, just one base pair. So I was working in the area of hemophilia, and it could take just a mutation at just one point to make the whole thing not work, and everything is broken. So a very small change is not the same as unimportant. It could be unimportant, it could be important. It could have large consequences or small ones. So if you change something through these methods, you mustn't forget you can always use these again. So if you don't regulate, the genome of one organism could systematically be changed and without the need of any approval process. Now, for me, that means that basically we have a circumvention of safety processes, and everything could be done with CRISPR-Cas. So for me, that means points to consider are: is it breeding? Is it really breeding what we are talking about? Breeding to me means evolution, where you can make take direct influence. But for me, this is genetic engineering. If genes are inserted, changed through external operational changes. So, is there a scientific consensus? Second item. No, there isn't, which it would be remarkable if there was, because science changes all the time. And at the same time, opinions in science change. So you cannot actually hope for a consensus to be found whether something is safe or not. But what is clear is that we have a need to know that something is safe, and for that I think regulation is necessary. And the other interesting point is the question how we deal with crises and emergencies. At this Rome panel that Professor Schiemann mentioned, it was interesting how to see the pressure that was being applied. Now, farmers do not have something that can make them adapt to drought or floods. What they get is breeding techniques, and they're being told that no regulation should take place to solve our problems. Now, simply this is something we could discuss for days on end, because what we need are crisis-resilient systems and structures to be more protected against crises, and we have changes on a population level. So we shouldn't produce a single seed that is uniform. We should have a certain breadth to be able to resist, and to have things that cancel each other out or support each other. And what we are doing is a techno fix rather than having resilience. We'll have to see how overall we deal with these things, because we need a broad debate. We have so many different solutions that we could use, depending on the way we want to face the future and engage with the future. Thank you. Good evening. Now, for legal experts, genetic engineering is much like pornography. Why? A judge of the US Supreme Court, Potter Stewart, said pornography is very hard to define, but as soon as I see it, I recognize it. So why is this important? Why is the question whether it is genetic engineering or not important? The different consequences that arise are the different approval procedures. You have the Seed Act in Germany, you have the gene law in Germany. Costs are different, it has been said. But also labeling is different, labeling obligations. The changes to seeds do not have to be labeled, and then there is a separation, obligation to separate. GMOs have to be separated very strictly to avoid mixing, because you otherwise cannot distinguish these genetically. Now, I've given you I've brought here the definition in our German gene technology law what a genetically changed organism actually is. It is an organism, with the exception of humans, whose genetic material has been changed in a way that will not occur under natural conditions through breeding and natural recombination. So that is the definition in the Gene Technology Act. And if in Europe you talk about environmental law or economic law, which has, of course, different overlaps, you have to talk about European law as well. And as you will see, that is similar. You have the release directive, which applies here, which says that a genetically modified organism is an organism, with the exception of human, whose genetic material has been changed in a way that is not possible through crossing or natural recombination. They've simply copied this, it seems. Now, I will not go through all of this. We have the distinction between different methods. You can imagine if you, for example, put a dog and a chicken into a cage, that will not work. If you insert different individual genes, that will kind of point to genetic engineering. But the thing is, to summarize what I have here, the procedure has to be made up in a way that organisms are produced that could not be produced naturally. So this refers back to the natural production or reproduction. Now, the release directive, which we have seen, is slightly more open at this point. You'd have to ask why. There is a sequence that says among others. So there's this talks about natural reproduction, but that is not comprehensive, because there is a second part which does not refer to natural production anymore. So you could come to the conclusion that this could or could not be genetic engineering. I'll leave this open at this point because as legal experts, we don't stop at this point. We ask. We have to imagine who wrote this. Some bureaucrat, a legal person like me, who may have this pornography comparison, asking themselves how to put genetic engineering into law, and who cannot include developments that took place later. So what does a legal person do when faced with a new technology, when they see that the law is much older? That person will not say that the law has to be applied 100%. Maybe they will look for an analogy, or they will have to develop the law, which is more or less the same. If we are developing law, we have to develop it in a way that is analogous to the potential threats that may have arisen or that may relate to the previously existing law. If you find that they have a gap in the law because technology has changed, so you have to close this gap, but you have to refer back to the threats that there are and see whether the threat potential is the same. So at this point, you would have to regard the risk potential and the thing that is to be protected. Now, open bracket, I heard that if I apply the Gene Technology Act, I need to conduct a risk assessment which is much more expensive than you would have to do in the Seed Act. You have to have a risk assessment there, but it's much less complex. If you now regard what the actual aim of these technologies are, the objective, and paragraph 1 of the Gene Technology Act is to protect the environment and its interactive character, and with regard of ethical values, life and health of humans, to ensure the opportunities to produce food and fodder conventionally, ecologically, and through genetic engineering. And of course, you have to keep in mind that you may have the opportunity to produce much more cheaply. Now, this refers to the environment as an interactive, as an interrelated system, and we've heard that these gene technologies are capable of being much more efficient, much more precise, and much more quick than conventional methods. So putting not the dog and a chicken, but two dog races into the same cage would be much slower than using these technologies. So this would be much more efficient, much more precise, and much quicker. So you wouldn't have a new organism every few years, but maybe every few weeks. And that, to me, seems to be the important difference to what you call conventional breeding. The whole process is fundamentally accelerated, and this acceleration must lead to the environment and its cause and effects and conditions has to adapt to these new organisms. Now, with risk assessment, you verify or you evaluate whether these new organisms have an advantage in selection. Now, with old genetic engineering, the change normally would mean that the organisms would be resistant against pesticides and herbicides. Now, that normally would not mean a selective advantage. If now we make individual organisms more resilient, more robust, then they do have a selection advantage. They could become, if they are more resistant, more robust, for example, in the rapeseed example, they could drive away the previously existing species. So the threat here is that the risk potential, as the legal profession would say, for these new methods would actually be higher than with the previous methods, because the way that the interactive ecosystem has changed. But you could, of course, say that there is no damage to the ecosystem. You just make certain plants more resistant, which is great. But even to prevent bad mutations is a change to evolution. You could say that the risk potential here is that the integrity of evolutionary development is disturbed, and that is the potential, the risk potential. And this has to be evaluated from a legal point of view, and I can only say that these old and new methods, to evaluate them legally, as long as legislators themselves do not regulate this. For example, in the Seed Act, you would have to perhaps insert analogous risk assessments into that, and these new methods of genetic engineering should perhaps be equated. So the Genetic Technology Act should perhaps be applied. Now, that's the first point. Now, the second, with that risk analysis, which has been previously made on genetically modified plants, a cost-benefit analysis is important. So this is a utilitarian kind of approach. That, of course, is always an individualistic kind of thinking. What are the individual benefits, the individual costs, which may be different to the societal costs and benefits. What you would have to do from an ethical and legal point of view, a societal cost and benefit analysis And that's where the benefits are really central. What actually is the benefit of genetically modified plants to society? It's always being said that we ensure the food supply of the world. Now, I read in a newspaper article recently that, and I quote, of the 7 billion people in the world, about 1 billion starve each day. At the same time, global food production would theoretically be enough to not just feed 7, but also 9, 10, or even 12 billion people. That was the result of this study, give or take 10% maybe, scientific imprecision. But still, it's enough for the 7, 8, or 9 billion that we have. It's a question of distribution. The use of genetic modification, to me, does not seem to be evident if you organize society reasonably. Now, that means you need a different kind of social organization where the benefit to society is considered as technologies are used or approved. That, I think, is a thought that would have to be considered urgently. Thank you. Okay, I would now like to give you an opportunity in the same order to react to what the others have said. All right, Mr. Professor Schiemann will start. So, first, in my view, CRISPR-Cas regarding the animal or human art, you could reduce errors drastically this way, but they're still too high to be defensible for changing human embryos, for example. Now, regarding plants, things are quite different. Plants are more error-friendly. You can afford to make mistakes. So all these technologies that we use, we permanently produce errors. But we have, as I said, we have a history of safe use. We have, we can live with these mistakes, these errors, and limit them. And we have all kinds of different opportunities in the legal field, food law, for example. So even if in this area we would have no regulation, we would still have regulation in all other. CRISPR-Cas is a relatively simple technology, of course, but I don't believe we can give it into the hands of individual farmers. That is somewhat too simple kind of thinking or speaking. Maybe this was for rhetoric effect, but it's clear that CRISPR-Cas still is high technology. You have to identify genes. You have to insert these genes under sterile conditions in the lab. And you have to have the breeder look at the results for many years to see how this plant behaves under environmental conditions. But because, of course, plants behave differently in the wild than they do in the laboratory. So in the future, it will still be not be the case that you give these technologies to individual farmers, but farmers can be involved as far as breeding is concerned. That, of course, is important. Now, where gene drive is concerned, this, of course, for plants is not really relevant. Well, Ricarda Steinbrecher disagrees. So what is being proposed and what's actually being done is quite different. Now, regarding insects, this is something that people are thinking about, but it's very clear that a whole range of more research is needed to actually apply this in practice. Of course, it's very clear that there's a whole range of levels to regard. It's a very important distinction whether I have an individual gene, the product that's being produced from it, or with the help of it, or a protein. So we have a high level of regulation, regulating various metabolisms, or whether I have a very normal gene that maybe leads to starch molecules branch or do not branch. So you have to look at individual cases and let's see what effects on which levels a mutation can have. And even with classic breeding, since Mendel's results, everything depends on individual mutations. And if you look at conventional breeding again, you do not have gene technology legislation. So there are a whole range of other mechanisms, and we cannot even conventionally induced changes are based on individual mutations. So off-target, very clear, CRISPR-Cas has this issue, but there are various ways for optimizing, and what all this, of course, will never lead to off-target effects being excluded completely, but what's important is that as we view the context of classic, conventional and other modern methods, we have to take that context into account. So regarding the legal statement from Mr. Fisahn, I would ask, what is natural? I think the more we have this continuum of what we had 10,000 years ago and what we do today, the more we look at this, the more we have to ask, what is natural? Gene transfer is natural. The occurrence of foreign genes in plants is natural. You have numerous examples. A very clear one is the sweet potato that that is a clear example. So gene transfer, even across species boundaries, is something natural. Now, the question of environmental consequences, the question of you referred to rapeseed. I will point to maize instead. Now, regarding the use, I think that we all agree that that the statement that genetic engineering could solve the world food problem, the global food problem, is complete nonsense. It is very clear that a single technology can never solve a global problem, but genetic engineering, together with a whole range of other tools, including new breeding technologies such as CRISPR-Cas, surely is an important tool that for certain problems, together with other tools, should be used. Perhaps that's what I would like to remark. I see. My position is different. My view is different regarding the continuum, which is always being pointed to. Allegedly, genetic methods are in a continuum with evolutionary development, so therefore, basically, you shouldn't take them so much more seriously. And for me, a genetic engineering method is something very different. We know that nature changes in certain so-called hotspots, and if we include these changes in positions and cause those changes to propagate in a way that's different to self-selection, so that we have a new species, that it has nothing to do with the old way in which we selected and changed the genome. Now, fundamentally what is happening is that we do no longer try to develop systems that support each other, whether it's in agriculture, in health, in water management. What we're trying is to always say, oh, there's a single problem here, and have an idea and have a technology. And often you have technology is actually waiting for a problem, and now some people say, now here's a problem. That's where this is a solution for. So we're not actually looking at the fundamental problems, and ask how we should deal with them. But we have these technologies, and that to me is linked to this. And however, the other point, the product versus process, and that's why I mentioned this also, Mr. Schiemann. If you look at the way when we negotiated safety protocols, it was said that we need one because genetic engineering makes changes that leads to other risks, new risks, not because we can insert them, but because the processes as such have consequences too. So if we have a mutation by inserting or delivering gene sequences, then we have to add this to the process. That's the difference. That's why I mentioned this. I know that the pressure is quite high currently to go away from the regulation of the process and just look at the product. That's what TTIP negotiations are saying, and the US position is for that other kind of regulation. But I consider it essential that we still that we trigger a process that looks whether something is safe or not, as based on the process. If you also say that there should be a new regulatory process looking at the product, then of course that is fine. Maybe we should look at the products too. We can do that, but we need, which you said yourself, we need a case-by-case consideration. It should be looked at on a case-by-case basis, and that's what we have to demand from legislators, so that we shouldn't release things that pose a risk or can cause a damage to the environment, health, etc. The history of safe use was mentioned, and that is why something was excluded from the legislation. GMO legislation, because it was said that we've had this for such a long time, but that to me doesn't mean that it's all safe. It still needs... We have no history of safe use with CRISPR-Cas. We've only had this for 2 or 3 years, so we cannot say anything. And in cases like that, we still need the GMO legislation, we still need it. And that, I think, is all that I wanted to say. And also regarding the legal position, it's interesting. I read it up shortly. In Germany, legislation is different. to some details of the safety protocol, but the foundation of all of that, of what internationally has been agreed upon, is that world trade should follow the safety protocol. These are the definitions that will apply, and that is even a bit stronger than what you have mentioned. So that's what countries have to follow, and therefore the Commission, the EU Commission, seems to be sitting on an uncomfortable fence. There's the TTIP debate. At the same time, they know that legally and scientifically there are problems, and that's why I think we won't have something by the end of this year still. But we still have to discuss these issues. Now, regarding if with these new technologies you refer to the history of safe use, you could say, well, don't just refer to the process or the products, but what you have in the background is the comparability to conventional products, and that is a discussion that even with conventional genetic engineering was relevant for risk assessment. Comparability to old products. There are products that are similar, and what was said is that this is the effects on the ecosystem. I talked about the interactive character. I talked about breeding, the debate whether something is breeding or not, because that's what the legal definition has. I said that the legal debate may have been surpassed completely by new procedures, new methods. It's very clear that most things that you regard as natural aren't anymore. So you have a mix. We have to try and use new developments or put new developments into the existing context. I would make another remark on the continuum issue. The problem is, and that doesn't just apply to genetic engineering, but other areas too. We have enormous acceleration occurring at the present. Also regarding the change of ecosystems or parts of ecosystems, and that could lead to problems in the medium or long term. So we may have a need for deceleration. We may have to look at long-term consequences, because it's very difficult to see what the long-term effects are, because you may only see them hundreds of years later. Now, genetic engineering does not solve world hunger, but what else does it solve? I don't actually know what problems exist for which we need genetic engineering. Now, if Sandoz produces a new chemical that destroys plants, that is not a societal problem. The important point is that to ask this question, which societal problem we have that is being solved by a new method that we therefore have to use, that is the question that we need to answer, and to include that in product approval. that's important. I would in particular like to mention the International Life Science Institute, which is co-financed by the chemical industry. Mr. Schiemann, a colleague of yours at the Food Safety Authority, the European Food Safety Authority, he was in the International Life Science Institute as well as the European Food Safety Authority when the current legal foundation that is still valid was being drawn up. Could you explain to us how this came about, how the European Food Safety Authority distanced itself from the Life Science Institute? Well, the European Food and Safety Authority came up with certain criteria, and the Food Safety Authority publishes questionnaires. So people that work in things like the GMO panel and other things have to submit these answers and identify potential conflicts of interest. And then the Food Safety Authority will decide whether there is a conflict of interest or not. And that happens with a lot of transparency, which I find very sensible. And the decision on whether a conflict of interest exists are being made by the Food Safety Authority.